So let's do this. Okay, so I said yesterday my favorite topic was base quality scores. So my second favorite topic is going to be variant quality scores. So here we go. So the idea is that we want to assign a confidence score to each of those putative mutations, right? Because you have tons of mutations that came out of the caller, and how many of them are right? You don't know. So you want to be able to assign a quality score to these variants so you can decide for yourself, maybe my project wants to use only the highest quality because I want to be so sure that I'm correct. Or maybe my project is worried about missing things and you just want to use all of the variants. So here we go. So we made all these mutation calls, and now we have to do tons of work to make those mutations analysis ready. So the purpose here is that, so by design, sort of these calling algorithms, the design of them, our philosophy is that we want them to be incredibly sensitive, right? So that you don't miss anything. And then it's up to the user to decide for themselves whether they want to be more permissive or whether they want to be more specific. And so as a result, the calls that come out of the unified genotype array and the haplotype caller, they're infested with false positives, right? Because we don't want to miss anything. It's a high sensitivity call set. So a ton of work has to go into filtering down these call sets into the real ones, right? And so for a long time, people were using these hand-tuned filters to sort of pare down the false calls, right? So you experts look at the data and decide, oh, this doesn't look quite so right, so let's create this filter to get rid of these things. But the problem with these hand filter approaches is that it's always different. Every time you come to a new data set, you have to invest the time, an expert's time, into creating these new hand filters. So wouldn't it be great if you could just learn those filters from the data and use the data itself to tell you which were the real ones and which were the wrong ones? That's exactly what we have here. So we have this procedure called the Variant Quality Score Recalibrator, which is going to learn from the data what filters it should apply. Okay, so, and in the end, we're going to build a model so that we can rank order all the variants based on their likelihood, and the user can decide for themselves how far down they want to go on that likelihood ranking list. And so they can trade off their project's goals appropriately. Okay, so what's the principle here? So the principle is that we've developed this diverse set of statistics that we're going to attach to every variant, right? So any number of things. So we've already talked about things like strand bias and quality score bias and all these things, right? So you can create any number of statistics and assign them to these variants. So these are things that you calculate on a per variant basis, right? And so what we noticed when you do that is that real variation tends to cluster together, and the false stuff tends to be outliers in these statistical space, right? And furthermore, what's very interesting is that most of these statistics, the way we design them, they cluster in a Gaussianly distributed way. It's very exciting. And so you can use a very simple Gaussian mixture model, since these are all Gaussianly distributed statistics, to model the data. And the variants that cluster together are going to be the real ones, and the outliers are going to be the false ones. So it's very simple. And so what it looks like in your VCF record is you have, Eric showed an example like this in the very beginning, where you have the chromosome and the start, and this is an A to G SNP, and this is the massive Phred scaled quality score. So the unified genotyper thinks it has 198 orders of magnitude of confidence in this variant. It's crazy. And these are all the statistics that have been assigned to this variant. So you have things like allele balance, and these are all cryptic names that you have to look in the VCF header to decode. But AB is for allele balance, AC is the allele count, so 3 chromosomes had this variant. This is the allele frequency, this is the depth, so there were 87 reads in this region. So maybe having excessively high depth or excessively low depth is a signal for being in error. There's tons of these things. Homopolymer run, mapping, RMS mapping quality, the number of mapping quality 0 reads, this thing called the quality score divided by the depth, so this number divided by the number of reads, things like strand bias, and any number of things that anyone can think of, right? And this is sort of a game now to sort of come up with new statistics to throw in here and see if they're useful or not, right? So there's entire fields, in fact, people spend a lot of time coming up with these statistics, these error sort of covariate statistics. And you can throw in anything to your heart's content, throw in here and see if it works. Okay? And so what happens is you can take those statistics and you can just plot them in whatever dimensional space you want. So right here, I'm taking two of them, the one that was called QD, variant quality score divided by depth, and the other one which was called SB, strand bias. And I'm just taking those variants and I'm plotting them. So now every dot on this plot is a potential variant in your call set. And these are those two dimensions, right? And so I have the top plot and the bottom plot. So the top plot are going to be, you have this proposed call set. And the top plot is all of the variants which were in dbSNP, right? These are the known variants. And the bottom plot, the green and purple things, are all the novel variants, right? So you have the known variants and the novel variants. Most of the known variants are probably real, and most of the novel variants are probably false, right? And what you see is that they make these beautiful clusters, right? So on the top plot, I'm plotting in yellow the things that were in dbSNP, but which weren't found in HapMap. Do people know what these databases are, I hope? So, and the red dots are the things that were in both dbSNP and in HapMap, right? And so what you see is that there's these beautiful clusters of the HapMap SNPs, and a cluster here, and then there's strangely these outliers that are in dbSNP but are not in HapMap, and they all seem to be like these outliers in this dimension, right? So they have, and you can sort of attach meaning to this strand bias statistic where negative numbers mean you have less bias and positive numbers mean you have more bias. And lo and behold, those outliers are in the space where you have low quality and more bias. So probably those are errors. Does everyone see how that happened? Okay, awesome. And so what we do here is you can use things like HapMap as training data and just learn a Gaussian mixture model. And so now these ellipses are actually the model that was learned by running this procedure. So this is a toy example from this paper that we published. And so we basically take these two dimensions, run a Gaussian mixture model over this HapMap data, and you come up with these 4 clusters, these 4 ellipses, and that's your Gaussian mixture model. You can take that model and now apply it to all of the novel variants. So you can just evaluate the probability of all the novel variants and set some threshold. And when you do that, that's where you get the green dots are now the variants which pass that threshold. And you see that they fall beautifully in these clusters here, and there's some in there which you can't see. And then the purple dots are the ones that fell outside of that threshold. They're out of the Gaussian mixture model. And so those are the potentially false novel variants, and those are filtered out of the call set. Does everyone see how that happens? Any questions about this?
When you apply this to other organisms, you can use the same filter that you have used for other organisms that do not have the gold standard.
Yeah. So there's a couple of things. So one thing you could do is, like you said, you could sort of take this model and try to just apply it to a new data set, right? And so that would be one approach. Another approach would be to sort of, in the same way that we could bootstrap a known set for the base quality score recalibration for these organisms that don't have dbSNP, we sort of bootstrapped a database. You could do the same thing here, where you could sort of make an initial call set, takes the ones that are the highest quality, and use that as your training data. And so the principle being that things that cluster together are the real ones, and the things that don't cluster together are the false ones. See what I'm saying there?
Yes, but to define the gold standard that I'm going to use to recalibrate using that, I have to use some parameters. You're just saying that maybe I can use some parameters, some of the variables that you have shown in terms of depth coverage or any kind of other things.
Yeah.
They're going to be, for sure, they're going to provide me the best.
Yeah. So you could. There's lots of things you could try, right? You could simply make the call set and take the ones that are the highest confidence, right? You could filter it down to the ones that you have extremely high confidence in. Those are probably real. As a set, those variants are probably real. And so you can use that to inform the evaluation of the rest of the ones which you're not so confident in.
But you wouldn't think it's a good idea just to use the same parameters that you have established for based on
So that's possible. I don't think that's how I would do it, but it's possible. The reason why is because you're sort of, every call set is different, right? So like if you're in one project, you're using like 20x coverage reads, you're going to have one distribution of statistics. But in the next project, if you're doing 100x coverage, or maybe you have 5x coverage of your organism or whatever, it's just going to, the distribution of statistics is going to be sufficiently different that you won't be able to just take the model wholesale and apply it. That's the only thing I'm worried about, right? So it's just a matter of whether or not you believe this distribution of annotations, whether they're going to be the same across these projects or not. If you think they are, then it's fine. Otherwise, I would be worried about that, right? Because you can't really see specific numbers here, but you could imagine like this negative 1,000 strand bias. If you have only 5x coverage, maybe you can't get all the way down to negative 1,000 strand bias.
Discard everything.
Exactly. So that's what I would be worried about. Is everyone good with Gaussian mixture? Yep.
So I guess I just want to follow up on the same point that
Yeah.
those genomes for which we don't have known, verified those variation sites. I suppose that what you're showing me here, the separation are mostly driven by the platform themselves, not so much by the genome variations.
They're driven by what? I missed
Like,
that.
plants versus human.
Oh, I see.
Is that because we are struggling with the same thing that we don't have, this known information for some of the genomes. So I was just thinking that we could extrapolate, of course, assuming the depth of coverage is about the same.
So I think, so this issue of organisms not having this high quality database of known polymorphisms, that's sort of a sticking point that comes up frequently, right? And so I think, but there are lots of strategies that need to be explored here. So what I think people in this situation should do is just simply take SAM tools, take GATK, take your favorite snip calling, call your variants with all these various methods, and the ones that are concordant, use that as your database of known polymorphism. Because the things that are seen by all these callers, they're most likely real. All you really need to do is come up with a set of variants that you have high confidence in. And there's any number of ways to do that, right? You could very stringently filter the call set. And the ones that are left are the ones that you have like just extreme confidence in. They're probably real. That you could assign a confidence of like 99.9% or something, whatever you wanted to that set of variants. And then use that as training data for base quality score recalibration, indel realignment, for variant quality score recalibration, all these things, right? So it's just a matter of like bootstrapping this database in whatever way you want to. And that's a research project of its own. People can do whatever they want. They think it's good. But I think that a strategy like that is how that community could move forward and use these tools.
To use something like most of these variants could be like transitional versus using like last
So so I guess I'm extrapolating from your question, but you're afraid that you're biasing the mutations?
So in general, you should see more, transitional versions. So I was wondering maybe that would be something to help.
Oh, I see. You're meaning as a statistic. Oh, yeah, sure. That's a good idea. Yeah. So you could try to add things like any sort of functional annotation things or any sort of biological interpretation that you wanted of the data and use that as a covariate for error and see if it worked. In general, when we've tried that, our group in general is we have less biological training, I would say. And so when we've tried to do that sort of thing, it hasn't worked out so well. But that's probably just because we don't know what we're doing. If someone who knew what they were doing tried to use those statistics, they could probably do a good job with them. Anything else? That's what makes this system so exciting, though, is because people can use any statistic they want. Anything they come up with, they can just throw it in and try it and see if it works. And we've had huge success with like 1,000 Genomes Project of collaborating with these people all over the world who want to everyone wants to contribute a new idea for a new statistic. And we just throw it in, try it out. And we can determine in like 2 hours whether it's a good idea or not. It's just so easy, and it's just so collaborative with all these people. So we've had a lot of success of just throwing in random annotations and seeing if they work. So we've been able to leverage, 1,000 Genomes Project and things like that.
With all these new SNPs being discovered, when you do a new whole genome sequencing, what % of high-quality novel variants would you expect to see at this point?
Yeah. Okay, so that's a question that requires like a very analytical answer. So I really have no idea. I mean, the right answer is that we've used this procedure, like Guillermo was mentioning before, that we've done this like extensive validation project through the 1,000 Genomes project, and when we did that, the SNPs had a concordance rate of what was it, 99.5%. So the false positive rate of 0.5%, 0.5%, which we were very happy with. So that's really the only answer I have. So the only like validation of these sort of methods that we've ever done is through the 1,000 Genomes project. And when we did that, this method performed very, very well. So sorry that's not quite so analytic, but anything else? So this, I think, is my only mathematics slide. I just wanted to show you a Gaussian mixture model. So basically we have pi and the Gaussian. So this is a Gaussian mixture model. And we use variational Bayes expectation maximization procedure, which means that we have a prior over the mixture coefficients. And that prior is a Dirichlet distribution. And so what that does is it enforces sparsity in the model. So that's the reason why in my diagram here, you only have 4 clusters. Because what the model decided for itself, how many clusters were most appropriate using that sparsity constraint. So it decided that many of the mixture components weren't informative, so it reduced it down to only 4 mixture components. So that's the point of this prior. And then you have this other prior called this crazy thing, which all that does is it's biasing you away from having singularities in the data. It's not quite so important. I just wanted to show some mathematics. Does this make sense? Do people care about variational Bayes? This is the part that I enjoy. You guys probably care about the biology. I don't know. Okay, anyway, I'll show you how it works. So like everything, we have a 2-step procedure. You learn a model, and then you apply that model. So the first thing is called VariantRecalibrator. So you're going to build a Gaussian mixture model. This is where you're going to come up with those modeling parameters. You're going to have this model file, and then you're going to apply that to, you're going to apply that to the VCF, and what's going to come out is a new VCF that's been filtered at whatever level you decided to filter it at, right? So in the output, every variant's going to have this confidence score assigned to it, and then you've filtered it at some confidence level. So here's how it works. So so we're in the first thing. So you take in your VCF, the original VCF file, and all this training data that we talked about. You run VariantRecalibrator, and that looks like this. So where all of the other command lines that we've shown have been very simple, the VQSR is unfortunately not simple. This is our most complicated command line. So you have fun reading through all this. So you have VariantRecalibrator, you have the reference again, you input your VCF file, you specify all of your training data using this resources tag, which we'll talk about on the next slide apparently. And then you specify all the annotations that you want to filter, that you want to cluster with, right? So these are those statistics. So you say which ones of those you want to use, and this is usually a very long list, like 8 or 9 or 10 different annotations. You can tell it to run in SNP-only mode, indel-only mode, or put them together. So sometimes maybe your SNP annotation distribution is different from your indel annotation distribution, so it wouldn't be appropriate to model those together. But sometimes those distributions are sort of unified, and so you would want to model them together. So it's up to you to decide. And then you have all these other various files. So you have this is the output of this procedure. So you have the recal file, which is just a list of variants and their confidence scores. And then you have the tranches file, which is going to show you all the like various ways to filter the data and the resulting truth sensitivity if you were to do that. And then you have beautiful plots that you can look at if you'd like. and that's specified through this. So it writes an R script file and then calls R on that R script and creates the plots.
Have you tried to generate the same kind of result but across several organisms just to see if actually the parameters they actually got affected by
Yeah.
peculiarities, by characteristics of organisms, or besides that, check the distribution of those variables for different depths of coverage.
Yeah. So the answer is no. We only work with human data in our group, unfortunately. So
What would be that actually they do not change? So maybe there is no reason.
It's
to
possible.
recalibrate.
I think it's more. It's not a function of your organism. It's more a function of your project design, right? Like if you're doing like a 20x coverage project versus a 100x coverage project, then I would expect those distributions to be quite different.
But maybe you can recalculate it based on the depth of
Yeah.
coverage. You just have already a priori the depth of coverage. And
Yeah.
Just an offset is going to happen.
It makes sense. We haven't done anything like that though, but it makes sense. Okay, so this is probably the most complicated command line that you'll see in the whole session. So hopefully it doesn't overwhelm you too much. I promised that we would talk about the resources, and so this is how you specify all the training data. So it looks like this, quite complicated. So every line is a different database. So here we're using the HapMap database. We're using a file from the 1000 Genomes project, which is this Omni 2.5 million SNP chip array. So we're using that as training data, and of course we're using dbSNP. So what happens here is you're specifying how you want to use that data. So there's 3 different tags. There's known data, training data, and truth data, right? So for each of these databases, you're going to say, should I use this as known data, should I use it as training data, or should I use it as truth data? And those mean 3 different things to the algorithm. So let's go through them in order. So known actually isn't used by the algorithm at all. It's just simply when you output statistics, you want to stratify it by known or novel rate because people care about this stuff. That's how they sort of QC call sets, right? They want to know what was my dbSNP rate or what was my transition to transversion ratio in dbSNP sites versus non-dbSNP sites. So all that known tag is simply used for outputting qualitative little statistics for you to look at. Okay, so not quite so important. Training data, of course, is the important one. These are going to be the variants that you actually use for training data in the Gaussian mixture model. So any variant that overlaps a database that's called training, where training equals true, those variants are going to go into the Gaussian mixture model for building the model, right? So in this example, we're using all the variants that overlap HapMap and all the variants that overlap this 2.5 million SNP chip array from the 1000 Genomes project. Those variants are going to be used as training in the Gaussian mixture model. And so that's specified by saying training equals true. Make sense? Okay, so the final one is called truth data. So you can say truth equals true or truth equals false. And so this is for when you're done training, you want to decide what level should I cut the variant call set at, right? Do I want to have truth sensitivity of 99%, or do I want to have truth sensitivity of 99.999%, right? Do I want to be very specific, or do I want to be very sensitive? And so the way to make this trade-off is to say, what percentage of sites that overlap the truth set have I retained in my call set, right? And so for this example, we're using only the HapMap sites as truth, right? So we say truth equals true for the HapMap, and then the default is like 99.9%. So you cut down the call set so that you've retained 99.9% of the calls that were originally in HapMap. Does everyone make sense how that happens? Okay. Right, so then the output of the variant recalibrator procedure are these files that we talked about plus the plots. And then you're going to simply feed those things in with your original call set and run this apply recalibration procedure. And then what comes out is your final call set with confidence scores assigned to every variant. And so that's very simple. Actually, it's just apply recalibration. You feed in all those files from the previous step. You feed in the recal file, the tranches file, and the output is going to be, this is now your analysis-ready VCF file where everything's good to go. And then you can specify where you'd like to cut the call set, and this is the true sensitivity filter level. So in this example, it's filtering at 99% of the variants that were found in your truth file, right? So this is, you want to keep 99% of the HapMap sites. You can set that to whatever you wanted, 99.999 or however you want to do it. Okay. And then, of course, the output is every variant annotated with its LOD score from the model. And that is it. So what comes out of this procedure is not only this call set, but also these mixture plots, so you can decide if the procedure worked well. So one way to decide is by looking at plots like this. So these are the model plots that come out of the VQSR procedure. And so what you can see is that you have these statistics now. This is just two statistics that we use for this example. It's the mapping quality rank sum test and the read position rank sum test. And so what you see is that the model learned that being in the center of these distributions is good and being on the outside of these distributions is bad. So that's a good thing. It learned that it's a Gaussian distributed distribution. That's awesome. And so now it's showing you which variants it kept and which ones were removed. It's showing you which variants were used for training and which variants were used for this negative training. And this is now showing you which variants were in dbSNP and which ones weren't, right? So the ones in dbSNP were in this distribution, and there were a lot of novel things that were out here, and those were probably the bad ones, and those were filtered out. So looking at plots like this sort of give you a sense of what happened to your variants, right? How they were distributed, which ones were kept. So this is one way to QC the procedure and decide if it's working well, is by looking at plots like this. Does this make sense? Does everyone feel like they could run this procedure and look at a plot like this and decide if it's working?
So you also generate some RC curve-like plot. For example, because you have negative case, you have this
Yeah.
case, and then you have a
Yeah, so in addition, there's this plot called, I think it's called the tranches plot. And so it's an ROC curve, a ROC curve, and which would be true sensitivity on one axis. And then we use, as a measure of the false positive rate, we use the transition to transversion ratio of the novel calls. We use that as a proxy for false positive rate. And so from that, you can make a ROC curve of the, using the LOD score. And so that comes out as well. I'm just not showing you here because it's just a line. It's just a standard ROC curve, so I didn't show it. But does that make sense?
And because before that we used, for example, the sequence depth filter, genome filter, but then, as you said, all these things are incorporated into this score.
Mm-hmm.
So does it mean that we only need to select a certain cutoff score?
Yeah, exactly.
We don't need to.
So the question is about what about all these things like depth filter and genotype quality filter and things like this? So the beauty of the VQSR is that it's putting all this stuff in aggregate into one statistic. And so you can simply cut along that one dimension now. You don't have to worry about depth greater than 5 or less than whatever, or genotype quality less than some number. It's all of that information is incorporated through the Gaussian mixture model into a single score, which is the probability of being real. And you can decide for yourself how you want to cut along that score. So in that way, it sort of puts the power into the hands of the user. Does that make sense? Yeah.
So based on your experience, what kind of variable should put in the
I see. So the question is about which statistics should we use and which one should we not use?
and you can put everything.
Yeah, you can put everything you want in there.
That should be some recommendation.
Yeah.
Experience.
Yeah, so we have this beautifully put together website which has what we call the best practices recommendations, right? And so in there, we have tons of recommendations for which statistics to use based on project design, and also which files to use as training data and et cetera. And we actually provide all of those files, all those training data files in what's called the resources bundle, which you can download. And so Basically, there's like recommendations based on whether you're an exome project or whether you're a whole genome project. You know, when you're an exome project, maybe some of the statistics aren't quite so important, so you don't use them. But if you're a whole genome project, maybe they are. So there's a few like subtleties in there, but all those details are in a best practices documentation, which we have on the website. So you can, and all those are incorporated into like, you know, the production process at the BroadE. So if you get production exome calls from the Picard process, those best practices recommendations are incorporated in there.
I mean, could you use genotype quality? Because isn't that a per sample measure as opposed to a per cycle?
Ah, you're right, you're right. So you're absolutely right. So genotype quality does not go in here because these are per site annotations, like you said. So you're absolutely right.
Why do you have to build your own model every time you've got a new data set? I mean, isn't it possible from all your extensive experience with thousands and thousands of things that you can get a sort of set of parameters that are best parameters and you just use those?
Yeah. So what we found, so the question was like, why do you rebuild the model every time? Why can't you just learn an awesome model and just use it every time? So what we found is that the data is just too variable to do that, right? Every time you run your project, just the distribution of statistics is just so different that it made more sense to learn the model from the data every time. In the same way that we do for the base recalibration procedure, we're learning a model there for every single lane of sequencing data. And it's different. Every time you turn on the machine, just everything is just so variable that we found that you really do need to learn these filters from the data, and they need to change every time for every project. It's just too variable is the answer.
On that training arm, so is this using training data that was just the AppMap?
So for this example, it's using what we have here. Oh, it's way back here. So it's using HapMap, but it's also using this Omni SNP chip array, which is this 2.5 million SNP chip array. So it's using both those as training data.
So just what are the purple dots on the training?
Purple. Oh, so the green dots are the ones that overlap those databases, right? So they're in HapMap or they're in the SNP chip, and the purple are simply the variants which were the furthest away from those training sets, right? So it's like you have a positive, like you have positive examples from the training data, and then it's just simply evaluating all those variants and saying which ones were the furthest away from that model, and it's coloring those as purple. It's very simple, actually.
So those dots are actually coming from your data.
Yeah.
The data that's overlapping with those databases.
Well, so the purple dots are not overlapping the databases, but the green dots are. Yeah. So every time I'm showing a picture with dots, those are the variants in your input call set, in your raw input call set that came out of Unified Genotyper or what have you. Yeah. It's just some of them are colored based on their status, right? And so the green ones happen to overlap the database. The purple ones just happen to be far away from the databases. And you'll see here, like for example, there's this purple one, but it's in the cloud of green dots. And that's because it was an outlier in some other dimension that I'm not showing, right? Because this is like probably 8 dimensions were used in this example. I'm only plotting 2 of them here. So there's probably some other dimension like strand bias or something where this variant is perfectly fine in these 2 dimensions. Read position rank sum test and mapping quality rank sum test is perfectly fine if you only looked in those 2 dimensions. But if you use strand bias, it is biased, and so it should be filtered out. So
why isn't purple just everything that's not in HapMap?
it's because so there are some real things which aren't in HapMap, right? And so you want to keep those. You want to keep those in your call set, right? Does that make sense? So like you have your medical sample, you sequence it, you call your variants, and of course some variants are novel, not seen before. Those aren't in HapMap, but they're cluster with the variants that are in HapMap, and so those are the real ones probably, and the ones that don't cluster with the HapMap are the false ones.
But I'm what I'm saying is that in this lower left plot,
Yeah.
green means found in HapMap. Doesn't purple just mean not found in HapMap?
No, unfortunately it doesn't. It just means, it's just, what I'm plotting here is just, it's really just like the lowest 5% of variance. The 5% of variance that were furthest away from your training set. That's all I'm trying to show here. It's really just, the detail isn't quite so important. It's just what I decided to plot. It's just what I found is useful for deciding if the procedure is working well or not. I just wanted to highlight the variants that are so far away from your clusters to be called as obviously bad. That's all it is. Does that make sense?
So can you comment on the prior of 15?
Ah, good. Very detail oriented. Okay, so here we go. We have the resources. We specified known, training, and truth. And then there's this final thing, which is the prior. And so what this is this is, so like everything, it's a Bayesian inference procedure, and we were evaluating evidence for positive or for negative. And so there we have a prior on what's the prior probability of any variant found in that database is true. And that's the prior. So 15 here means Q15. So you turn that into a probability, which is something like 98% probability. So so there's like this 98% prior on being true simply by being a member of HapMap. And it goes down. So Omni is slightly less high quality. So the sites on the Omni chip have a rate of being real at Q12 rate. And then we've shown you many times when dbSNP is not so great. And so we assign that a probability of Q6, which is actually quite low. Something like 87% of variants in dbSNP are real. And so that's encoded right there. So it's just sort of, I mean, we picked these numbers based on our experience of using these databases. If you had some analytical way of deciding what the real true positive rate of these databases were, you could put that right in there. So we're just these are our best guesses based on our experience with the databases. Their true positive rate. And this is not the true positive rate of the genotype, because of course these SNP chip arrays are very accurate. They're accurate to a rate that's much higher than Q12. It's the probability of a site of the variant being variant at all, right? Not for any particular genotype being real, because the chips are real. They have a true positive rate of like Q30 or Q40 or something. It's very highly accurate. But it's the probability that the site is variable at all, given that the chip said it was variable. Does that distinction make sense to anyone? Okay. Thank you for asking. It's a very important detail.
Sorry, this is a basic question, but how do you convert all these sources?
Oh, so it's a Fred scaled value. So 10 Q10. So it's log 10 of the whatever, minus 10 times log 10. So Q10 means 90%, right? 10% error rate. And then Q20 would be 99%, and Q30 would be 99.9, and it just goes on and on. So we treat every probability as Fred scaled. That's why you specify it in here as Fred scaled. Okay, so let's see if that's it. So what did we say here? Oh yeah, we looked at model plots. So we finished this, and now we're down to these, and then that's it. Thank you.